Okay, like I was saying, Dr. Leasure is a professor at the University of Houston, and she and I crossed paths at the University of Texas at Austin, where both of us were graduate students in the psychology department. And at that time, I was frantically trying to finish a dissertation, and she was a bright young graduate student and I tried to not jade her about how horrible the last years of graduate school were. So I'm glad I didn't scare her off completely, because she's been an excellent colleague and an excellent researcher. Other things I want to tell you is after she got her PhD at the University of Texas at Austin, Dr. Leasure did a postdoctoral fellowship with Rusty Gage at the Salk Institute. This was a really important lab, and Leigh was one of the people who was doing work to show that, yes, adult brains can create new neurons, and I'm sure she'll tell you a little bit about that work as well. Currently, her research focuses on the potential of exercise to heal the brain, which is what you're going to hear about today. She has been funded by the Cancer Prevention and Research Institute of Texas to study exercise benefits for the brain after damage due to pediatric radiotherapy, which I didn't know. That's really cool, Leigh. She has also received multiple grants from the National Institute on Alcohol Abuse and Alcoholism to investigate exercise-driven brain restoration after alcohol damage. So I am really excited to have Dr. Leasure here to tell you about her work. And from what I remembered, I think she's going to also talk a little bit about her pathway, given all of those students that are in the room. The last thing I'm going to say, this lecture is brought to you by NeuroSci, which is a neuroscience honor society. And Lindsay and Maya, who's on the screen or on Zoom, they both applied for a chapter grant so that we could have this event. So congratulations to that. And with that, I will let Dr. Leasure start her talk.
All right. Thank you, Dr. Kozlowski. I just want to make sure everybody can hear me.
Is that volume okay back there? Yeah? Okay. All right. You're good, Leigh.
Okay. So thank you for that introduction. And I'm really excited to be with you all as part of your Brain Awareness Week and to support the activities of NeuroSci. I love that clever name. So as Dr. Kozlowski said, I am interested in exercise effects on the brain. I have been my entire career. And I'm not really going to tell you much about my career path, except to say that I had a very transformative experience as an undergraduate in the classroom. I had no intention of majoring in psychology. I was going to be a biology major and go to law school. And then I took this fantastic class about the brain. And I was hooked. And so because of that, I've always made it a priority to include undergraduates in my research. And so during my talk, I'm going to highlight some undergraduates who have contributed really important stuff. So to start off with, if my slides will advance here, yes. I want to tell you about why exercise is good for the brain. So down in the right-hand corner of this screen, I have listed a couple of things that exercise does. So in purple are bad things that can happen in the brain. So for example, high levels of inflammation are associated with neurodegeneration. Oxidative stress is kind of like stress on the cellular level, like cells get stressed out just like we do. And it's not a good thing. They don't function very well when they're stressed out, particularly neurons don't function very well under high levels of oxidative stress.
I can't.
And excitotoxicity is, as the name implies, toxic levels of excitation. And so this also is something that can contribute to neurodegeneration. So if you're starting to get the picture that exercise is something that's very protective, that is true. It's also something that can really help to build the brain. So Dr. Kozlowski mentioned neurogenesis and making new neurons. And this is something that exercise really has tremendous power to do. So in the regions of the brain that make new neurons, which are very limited, the hippocampus is one region that does it. exercise will really increase the number of newly generated cells in the hippocampus. And excitingly, for people like me who are in middle age, exercise seems to be able to provide all these benefits for the brain, even when it is older. Last item on this slide, trophic support. Trophic support refers to what I like to think of as brain fertilizer. So substances like brain-derived neurotrophic factor have been called like miracle grow for the brain. So obviously it's a good thing. It's something you would want. And so exercise has the capacity to increase trophic support. So for all of these reasons, exercise is really a tool that can maintain brain health, or if there's a situation in which we need to restore brain health, it's really excellent for that too. So exercise has the capacity to increase neurogenesis and trophic support and decrease some of those detrimental processes that are listed there in purple. And so I study rats. I do have some human subjects research that I will tell you about if there's time, but most of my work throughout my career has been with rodent subjects. And so I'm running rats and mice in wheels, and I'm trying to make a translational leap into what's going on in humans that exercise. And so this is me finishing the Rock and Roll Marathon in San Diego, California, with the spectacular time of 5 hours and 17 minutes, which is about 12 minutes per mile, which, hey, I got there. And I think that all of these wonderful benefits for my brain were happening across all 26.2 of those miles and all the training up to it. Now, you do not have to run a marathon to get these wonderful brain benefits. So nowhere close to 26.2 is required. And people often ask me, well, what's the best type of exercise for the brain? And I say, the exercise that you will do. So if you don't want to run a marathon, you don't have to. I heard Dr. Kozlowski mention yoga. That is good for the brain too. So fortunately, since I'm trying to make a translational leap from what's going on in rodents to what's going on in humans, it turns out that decades of research at this point tells us that what exercise does for the rodent brain, it also does for the human brain. And so while this conversation is never likely to take place between two rodents, hey, whether or not they have an exercise bike or an exercise wheel, the things that are happening in their brain when they exercise are very much parallel to what's going on in our brains when we exercise. So over the years, I've used exercise in rats and mice as a model for how we might help humans who have a situation in which the brain's health is not optimal. And there are a wide range of possibilities out there. So I'm just going to tell you about two of them that I have focused on during my career. So the first one is exercise benefits after pediatric radiotherapy. So pediatric radiotherapy is a term that means that a developing brain, a child's brain, has been irradiated for therapeutic purposes. And so generally this happens for cancer treatment. And as you might imagine, when you irradiate the developing brain, that has some pretty serious implications for the brain's developmental trajectory. So it's one thing to irradiate a fully adult brain, at least the brain is fully formed. But when you irradiate a developing brain, you change the way it develops. And so there's a lot of research from survivors of childhood cancer and childhood cancer treatment with radiotherapy that shows that they have cognitive problems, affective problems into adulthood, and that their quality of life is really very different than that of people who didn't undergo radiation as children. So for example, they might be less likely to go to college or less likely to get married. And so given that exercise is a relatively inexpensive, easily accessible way to either maintain or restore brain health, we wanted to see if it could be restorative after early radiation of the brain. And so I did this in conjunction with my colleague, Dr. Wally Gaber at Baylor College of Medicine. In fact, I knew nothing about pediatric radiotherapy until I met him. And then my postdoc and graduate student did much of the work that I'm about to show you. So just to kind of give you a picture of what we were trying to achieve here, radiating the brain is associated with all these bad things. So increase in inflammation, stress, excitotoxicity, decreased neurotrophic support, and neurogenesis. Exercise has the capacity to reverse all those effects. And so what we did was take very young rats, so 5 weeks of age, that's very early adolescence for a rat. And so our experiment had 4 groups. We had 2 sham groups that got no brain irradiation. And we had 2 that got CRT is just stands for cranial radiotherapy. So these were groups of rats that were irradiated at age of 5 weeks. And so at a very young age, their brains were still developing. After that, the animals were either sedentary or exercised for 7 weeks, starting at about 7 weeks of age. So this is still adolescence for the rats. And so they had a good long time to exercise from adolescence into adulthood. And then thereafter, for the rest of the experiment, they were sedentary. So after age 14 weeks, there was no exercise. And this is going to be very important in a couple of minutes. After they finished exercising, we trained them in what's called the 5-choice serial reaction time task. And so this is a task, I'll tell you a little tiny bit about it, and it's not going to sound very hard to you, but please take my word for it. It is a challenge for rats and mice to do this task. So the task is basically that they're sitting in a little chamber and there are 5 apertures at one end of the chamber. And here's the task. When a light comes on in a particular aperture, the rat has to go and poke its nose into the hole where the light was. Can't poke it into this one, can't poke it into this one or this one or this one. Those would be incorrect responses. So you got to poke your nose in the hole with the light. And when that happens, that's a correct response and you get a little chocolate pellet in your food magazine. So you have to turn around, get your chocolate pellet, eat it, and then turn around and start monitoring these apertures again so that you can stick your nose in the one that was lit. And so you might be thinking, well, that's not very hard. And indeed, rats learn to do this, but they do make mistakes, and they tend to make more mistakes the shorter the length of time that the light is available for. So if the light is only there for a fraction of a second, it makes the task very difficult. And the other thing that makes it difficult is when we increase the delay between trials. And so when we increase the delay between trials, what rats will often do is start just sticking their nose into any aperture, hoping that that will magically produce a food pellet, and it doesn't. And so we call those premature responses. And so those premature responses are indicative of impulsivity. Impulsivity is something that is linked to the functioning of the prefrontal cortex. And so that was exactly what we irradiated in the rats, was their prefrontal cortex. And so what we were trying to see was, would cranial radiotherapy produce impairments in the 5-choice serial reaction time task? And could we sort of attenuate those impairments if the animals had had exercise? So in addition to that, we also did some neuroimaging. This is a little rat MRI, and we were looking at things like overall size of the brain between groups. And so what we found, there's a lot going on here, so let's walk through it gradually. I want to point out, very important, that all of these tests that I'm going to show you were performed at least 6 months after radiation, so after the cranial radiotherapy, and at least 4 months after exercise. So remember, by the time they were ever tested, it had been a very long time since they had exercised. So we're really looking at the effects of exercise during development on cognitive function in adulthood. And so we had been hoping to find that cranial radiotherapy, or CRT, would produce impairments in the 5-choice reaction time task. And really, we didn't. But what we did find is, in my opinion, kind of far more exciting, that 6 months after cranial radiotherapy, 9 months and 12 months, later. We found that exercise reduced impulsivity. It reduced the percent of premature responses in all of our exercised animals. Didn't matter if they were irradiated or not. Exercise during development helped animals reduce their impulsive responses in adulthood. And again, I find this really exciting because it had been a long time since there had been any exercise. So this is like a developmental stamp that exercise is putting on the brain. Now, what does that actually look like at the neural level? That's a completely separate question. And so we tried to answer that with our neuroimaging. And so we were looking, for example, at total brain volume. And we found that radiotherapy, cranial radiotherapy, reduced brain volume, but that in exercised animals, so there was a main effect here, so that exercise increased everybody's brain volume, regardless of whether or not they had had cranial radiotherapy. So what we took away from this was that exercise during development has some definite benefits for the adult brain. And I think this is really interesting, especially in light of the increasingly sedentary behavior of all Americans, but for sure, including children and adolescents. So from there, I'm going to switch gears to a different type of brain damage, and that would be damage from binge alcohol consumption. And so the funding that I have now from the National Institutes of Health is basically to look at the capacity of exercise to aid brain repair after damage from binge alcohol. And so a little bit of background on this. And AUD is just an abbreviation for alcohol use disorder. Exercise is used already and has been for decades as sort of an adjunctive treatment for alcohol use disorder. And the reason is that it has the capacity to reduce comorbidities. So that's a fancy way of saying that exercise helps with things that go along with alcohol use disorder, like anxiety and depression. And so exercise can kind of help alleviate those conditions a little bit. And it's also an alcohol-free way to spend time. So this is sort of the rationale for why exercise is used as an adjunctive treatment. And adjunctive means that it's something that's done along with other treatments for alcohol use disorder. My question has always been, can exercise repair the brain after it's been damaged by alcohol? And so to look at that, we use a rodent model, rats. Rats, I will tell you, are not dumb enough to binge drink. And so we have to give them alcohol. So this is a feeding needle. And so we put that down the esophagus into the stomach and give them a dose of alcohol. And that dose happens to be 5 grams per kilogram. That is a lot of alcohol, but we did go through a fairly lengthy process of seeing what blood alcohol level we would get from that dose and verifying that it would be compatible with a blood alcohol level that you would see in a human that was binge drinking. So incidentally, if you're wondering what binge drinking is, the sort of informal definition is that it's about 4 drinks in a 2-hour period for a female and about 5 drinks in a 2-hour period for a male. The more formal definition is that it is a pattern of alcohol consumption that brings the blood alcohol level above 0.08. So the bar is low for blood alcohol level in terms of what constitutes binge drinking. When people binge drink, they are taking their blood alcohol level way past 0.08. So for the rats, we give them a 5 gram per kilogram dose, and we do this on a Monday evening. They get Tuesday off, and then they get the opportunity to exercise in a wheel on Wednesday, Thursday, and Friday for about 2 hours. So, they're not exercising for 12 hours. They will run 5-6 kilometers in 2 hours. So this is really something that they like doing and they will readily engage in. And so this was sort of the layout of our experiment. On Monday, they would get their binge. Wednesday through Friday, they would be able to exercise. So half of the animals had binge, half of the animals then had exercise after they binged, and half were sedentary. And so the blood alcohol levels that we were getting from this were around 0.17. So about around 175 milligrams per deciliter, which in human terms would be a BEC, a blood alcohol level of about 0.17. And so it was compatible with what is being achieved by human binge drinkers, which was what we were trying to do. So this was good. We did this paradigm of binge on Monday, exercise Monday through Thursday for 11 weeks. There was nothing magical about 11. It just worked out that way. And that's a whole long separate story that I don't really have time to tell you about. What I want to tell you about is that at the end of 11 weeks, we harvested the brains and we sectioned them and we stained them for nu-N, which is a protein that's specific to neurons. And we were interested in counting the number of neurons that were present in a very special area of the brain, the dentate gyrus of the hippocampus. And so I'm tracing it here with my laser pointer. So this whole structure here is the rat hippocampus. And so the hippocampus is interesting for many reasons. First of all, it's associated with learning and memory. Second of all, it's very vulnerable to the effects of alcohol. So alcohol will damage the hippocampus. It's also one of the regions of the brain that can make new neurons. So this little V-shaped structure, it looks like a V on its side. The dentate gyrus is the place where new neurons can be made. And so binge alcohol sort of suppresses the ability of the dentate gyrus to produce new neurons, and exercise does the opposite. So what we were doing was looking to see how many neurons would be present, be left over in the hippocampus after animals had binged and or exercised. And so this is what granule neurons look like at high magnification. And so we use software that allows us to set little counting frames over our dentate gyrus and then count individual cells. So it is a painstaking process. What you get when you are done counting all your sections is, generally speaking, about 1 million neurons in this hippocampal dentate gyrus. So we're measuring this in multiple sections, not just this one section, but multiple sections, maybe 6 from the front to the back of the hippocampus, and then using stereology to sort of estimate the total population based on that. And so when we do that, the estimated total population in a control animal that got no alcohol and is sedentary is about 1 million neurons. Animals that binged or got a binge dose of alcohol every week for 11 weeks had a significant, about 20% loss of neurons in the dentate gyrus. So the question, of course, is, well, does exercise help with that? And the answer is yes. So exercise was able to completely prevent that loss of neurons. And so the reasons for that are a long story. It has to do with both maintaining a certain level of neurogenesis, which with exercise, and also getting those cells that were generated by exercise to incorporate themselves into the existing hippocampal network. So in addition to looking at number of remaining cells, we wanted to look at the response of glia. So here we're looking at neurons. We also wanted to look at glia. And so we focused on microglia, which are essentially the brain's immune system. And so they get kind of upset when there's an ongoing adverse situation in the brain, which of course binge alcohol is going to induce acutely. That means for several hours after the binge, it's going to induce a negative state in the brain, and microglia are going to respond to that. And so they respond in various ways. For one thing, they make more of themselves. So they're sort of like your first responders. And so if there is a situation in which, the brain is under duress, like from alcohol, microglia will help out by, for one thing, dispensing neurotrophic factors to neurons to kind of help them withstand the adverse circumstances, which in this case would be alcohol. And so what we saw with our animals that either got binge and or exercise or remained sedentary was that for total microglia, we didn't see a significant effect of binge on total microglia. We did see that exercise drove down the number of microglia. And so this can be interpreted in several ways. You could look at it as, well, the situation within the brain wasn't too bad. So we didn't need that many microglia. So in the exercise brain, we've got all this brain fertilizer circulating. And even when we add alcohol on top of that, we don't really see the need to have a lot of caregiving by microglia. The situation in the brain is just so much better with exercise that you could add alcohol onto that and not really have too much of a damaging effect. We saw the same thing with primed microglia. So primed microglia, this is a term that sort of means the microglia are active. So these would be called ramified. These over here would be activated. And so they would be in a state of needing to provide some sort of assistance. And so we did see that with the binged group that was sedentary, but again, exercise quelled the need for a microglial response. So from this, we took away that exercise counteracts binge-induced hippocampal damage. Now, there's a lot of reasons that it might do that. And my lab went through a period of time, so you can see we published this back in 2019. And obviously, there's been a pandemic since then, which has made research a challenge for a couple years there. But we went through a period of taking of directed hypotheses. We said, okay, well, maybe the reason that exercise helps to repair the damaged hippocampus is that, it's driving down cortisol levels. Or we had all these ideas about what the mechanism might be. So we really wanted to know, why is exercise helping to repair the alcohol damaged brain? And so what we ended up deciding after a couple of years was to stop looking at specific hypotheses and try a more generalized approach. And this is something that's very increasingly common these days. Everybody's doing it, so we decided we would too. We wanted to do an RNA sequencing experiment. So we used the same paradigm that we had been using to show exercise-based restoration of the alcohol damaged brain. One little twist, we added another day of exercise. So for our binge and exercise group, they're getting their binge dose on Monday, and they're offered the opportunity to exercise on Tuesday. And if you're wondering whether they're reluctant to do that, no, they're not. They are perfectly happy to get in the wheel on Tuesday and run. And what we were looking at was the transcriptome. And so I want to address what the transcriptome is here just for a bit. So DNA, your genes, your genetic code, your DNA codes for ultimately proteins. But there's this intermediary step there called RNA. And so DNA is transcribed into RNA, which is subsequently translated into proteins. So if you want to kind of get a global idea of what kind of changes are happening in the brain at the protein level based on exercise, you can look at what is called the transcriptome, because the transcriptome is essentially all the RNAs that are produced from DNA during a specific period of time. So we felt that if we were to look at essentially the transcriptome of animals that had been exercised and binged, we might be able to pinpoint genes that were involved in that exercise-based restoration. And so to do this transcriptome experiment, we did the same thing we'd been doing before with binge on Monday, Tuesday, Wednesday, Thursday, Friday exercise. We did that for 5 weeks this time. 11 was more than enough. We've learned that five will do it. In other words, 5 weeks of once weekly binging produces all of those adverse changes in the hippocampus, and they are remediated by exercise. So we did our paradigm for 4 weeks, and on the 5th week, we killed the rats right after their exercise on Tuesday. We dissected out the hippocampus, we extracted the RNA, and we sent it away to a company called Novagene to basically sequence it. And so once they sequence it, what you get from that are what are called reads, which I'll explain a bit in a bit. But I want to say that I have no experience with the pretty high level bioinformatics that you have to be able to do in order to actually interpret your reads data. And so I'm fortunate that Erica Mehrhoff and Marissa Ehringer at University of Colorado Boulder are also interested in the effects of exercise on the alcohol exposed brain. And so they really have been teaching me and Theo Andre, who is an undergraduate in my lab, how to process this data. And a little bit of a funny story about Theo. He had asked me at the beginning of last fall semester if I needed any help in the lab. He'd taken a couple of my classes and he wanted to learn more about neuroscience. And I said, well, I don't really have anything for you to do, but I've got this RNA-seq study and I'm trying to learn how to process this data. And I said, so you could learn with me. I can't teach you this. It's going to be, Erica and Marissa are going to lead me through this and maybe I can teach you. And Theo said, great, sounds good. And so what actually ended up happening was that Theo learned all the cluster computing and the R and all the stuff that you need in order to be able to interpret these reads, and far outstripped my level of learning. And so essentially now Theo is teaching me. So it works out really well for me. So what he's been doing in conjunction with Marissa and Erica is taking the raw read data and basically assigning, so each read is basically a transcript of RNA. And in order to know what RNA it is, you have to go and match it to the rat genome. And then you have to quantify the number of reads per gene. And so in order to be able to do that, we had to do, I had to learn cluster computing. And then after you get your actual read counts, which is the number of reads per gene, this will give you an idea of how highly transcribed a gene was. So in other words, if you have a lot of reads for a particular gene, it means that that gene was turned on at the point at which we sacrificed the animals. So out of all this, you get what are called differentially expressed genes. And so what we thought was going to happen was that we were going to have a set of genes that was changed due to exercise. In other words, their transcription would either increase or decrease due to exercise. And we thought we would have genes that were either increased or decreased by binge alcohol exposure. And what we were really interested in was genes that would be changed by both manipulations, because we thought, okay, well, these are going to be the ones that are actually involved in exercise-driven restoration. Well, as so often happens in science, that isn't at all what we found. In fact, what we found was that there were 806 genes that were changed by exercise, 7 that were changed by binge exposure, and 1 that was affected by both manipulations. And so that gene, it turns out to be a zinc finger protein, which is essentially a transcription factor that will determine the extent to which other genes are expressed. And so one of the things that we are doing right now is looking at what are the downstream targets of this zinc finger protein. So this was very disappointing. It was not what I was expecting at all. But hey, that is science. After you get your differentially expressed genes, another thing that you can do with your read count data is to submit it to what's called a weighted gene co-expression network analysis, or WGCNA. And so I will show you a picture of what that kind of analysis generates. It generates a lot of heat maps. And so essentially what you've got over here, this is showing the relationship between the genes. So the analysis spits out what are called modules. So here is one module. It's module number 19. And the modules are basically clusters of genes that are expressed in similar ways across all your samples. So it's a very agnostic approach to trying to figure out the big picture of what your manipulations have done to the transcriptome. And so every line in this heat map corresponds to a gene that is in this module. Every column is an individual sample. And so just looking at this, if you look at the right side of it, you see a lot more blue than you see orange. So all of the samples over here are from animals that were sedentary. All the samples on the right are from animals that were exercised. And so you can just look at this and go, wow, okay, a lot of things changed as a function of exercise. If you add the binge and control categories onto that, so in other words, all of these samples are from animals that never got alcohol and they were sedentary. These are from binged animals that were sedentary. These were from control animals that exercised, and these were from binged animals that exercised. And you can kind of look at this and go, yeah, okay, maybe there's a differential expression pattern across these 4 different groups, but overwhelmingly, it just looks like there's a big effect of exercise. And so what we take away from this, and we're kind of in the early phases of this analysis, but what I take away from this at this point is that exercise exerts a really powerful effect on the transcriptome. And this makes sense given all of the exercise benefits that we know it has for the brain. And so just in the last couple of minutes here, I want to tell you about another undergraduate in the lab who was sort of, looking at results that like this and going, wow, exercise really has a powerful effect, and binge exposure doesn't seem to impact that. So her name is Sarah Semmel. She's an undergraduate in my lab. She was interested, looking to see if we could see exercise effects, like if the effects of exercise on the transcriptome are so powerful, and if they're so powerful on the brain, would we be able to see effects of exercise on binge-related behaviors? And so this is a way of saying Sarah was interested in seeing whether exercise exposure affected hangover. So first, she had to kind of figure out, what does hangover look like in a rat? And so fortunately, we had recorded, we're all sitting there with our iPhones. After we would give the animals their binge dose, we would record their home cage behaviors. So we were looking at intoxication behaviors. We were looking, it's one way to verify that our manipulation has worked. Are the animals acting intoxicated? And then we had also started getting video footage of the animals on Tuesday mornings before they went into their wheels. And we were doing it in the morning because the lights go out in the morning. So our animals are on what is called a reverse light dark cycle. Rats are nocturnal. They're active in the dark. So if we want them to be active when we're active, we have to turn the lights off in their animal room during our waking hours. So we were recording our rats in their home cages on Tuesday mornings, right when the lights went out, which in other words is when they wake up, to see whether we could detect any differences in the animals that had been exposed to alcohol and the animals that had been exposed to alcohol and exercise. And so Sarah watched a lot of footage of rats doing their thing in their home cage, and she sort of settled on, well, I think maybe I could score 2 behaviors that might be relevant to what we would think of as a hangover in a rat. And so one of those behaviors is grooming. So you've probably noticed that your dog or a cat spends a lot of time fixing their hair. Rats do the same, only they're not really fixing their hair. They're sort of wiping their saliva over their head. This helps them with things like thermoregulation. And so a normal rat will spend a lot of time grooming. That's what they're supposed to do. So she decided, okay, I should look at grooming behavior and see if rats that were binged spend less time grooming, and rats that were binged and exercise spend just as much time grooming as control animals do. So she decided she would record time spent grooming, and she would also record time spent inactive, because when the lights go on, the rat should be very active. So these were the two behaviors that she decided that she would score. And so she's not even close to being done, and I'm not really sure what to make of her data, but I thought I'd show it to you anyway because it's kind of fun. So the picture looks kind of straightforward in females. So, female controls spend around 40 seconds out of 5 minutes. So these are 5-minute bins that we were looking at. In 5 minutes, they'll spend about 40 seconds grooming. Binged animals, less. With exercise, everybody starts grooming more. Okay, great. Looks like hypothesis right on target in the females. Time spent inactive, okay, great. We've got, very little inactivity time in control females, a lot in binged females. And I don't know why exercise increases inactivity time. This makes no sense to me, but it does seem to fix the problem in the binged animals. In case you were thinking, well, they were exercised, so maybe they're tired. No, I want to remind you that we are recording them before they ever get in their exercise wheel. So they haven't actually exercised since the previous Friday. The data is all over the place in the males. The time spent grooming data looks straightforward enough, but we've got this big uptake in inactivity in exercise control males, which I can't explain, and I'm not going to try. All I will say is that I really credit Sarah with making the ethogram of behaviors to look to quantify, and then to spend a lot of time looking at rats hanging out in their home cages. So I was going to tell you a little more about exercise and hangover, but I really don't have time to. Let's just get to the bottom line. In human subjects, exercise seems to reduce the experience of hangover. So that's kind of interesting, and my colleagues and I are preparing a paper on that. But to be time accountable, I will wrap up and say thank you to all of the undergraduate and graduate and postdoctoral students who have helped me with this research over the years. And I'd like to thank our funding from both the University of Houston and from National Institutes of Health. And I've cleared this with Dr. Kozlowski, but I thought it would be fun to show you a lab picture from a long time ago, when Dr. Kozlowski and I were graduate students in the lab of Dr. Tim Schallert. So Tim died a couple years ago, and I know Dr. Kozlowski and I still miss him, but his mentorship legacy lives on in both of our research and careers. So I will end there and see what questions you might have. Dorothy, I can't hear anything. You must have muted.
Can you hear me now?
Okay, can you hear us now, Leigh? Okay, so you missed me talking about how I look exactly the same.
And do I.
We have aged gracefully. Josh, you had a question.
Well,
Go ahead
I just
now.
wanted to ask if you could give like a quick summary of how hangover works in people and how exercise combats that.
Well, we don't know why exercise combats that. We could speculate that, it has something to do with reducing inflammation and, basically to put it very, very succinctly, all of the bad things that alcohol does, exercise has the opposite effect. So the data that I didn't show you, I will tell you, first of all the issues with it are it's retrospective self-report. It's undergraduate students at two big Texas universities just reporting on the number of drinks they've drunk in the last 30 days, the number of hangovers they've had in the last 30 days, and their typical number of minutes of exercise per day. And from that, you know, we're able to see that the more you drink, the more hangovers you have, but that is less true for people who vigorously exercise. We can't tell you direction of effect. So one of the things that I would like to do, and I have a new graduate student who's interested in doing this, is actually get undergraduates to report on a daily basis, maybe for 3 weeks, maybe if they will, if we'd probably have to pay them a lot to do this, but tell us just, you know, how much did you exercise today? How much did you drink today? Did you have a hangover today? And then we could really start to look at what's going on in a more fine-grained analysis.
We do have a question in the chat. This is from Dr. Lim. He says, I may have missed this. Is there a way to translate the running wheel time to human? Is it equivalent to light exercise or intense exercise?
Yeah, that's a really good question. To answer that, I kind of like to look at distance, time, and speed to kind of get an idea of how much this would mean in terms of human exercise. But to some extent, it's hard because rats don't get in the wheel and run on it for like half an hour like humans do on a treadmill. They get in and they hop out. And then they get in and again and hop out. So their total time might be, you know, 45 minutes to an hour, but that's not sustained exercise. Whereas, you know, humans on a treadmill tend to just get on the treadmill and slog it out for X period of time. So yeah, translating it directly is tricky.
I think that's the case with anything we try to do in animals is trying to figure out how to translate it from the rat to the human. But a lot of the work that Dr. Leasure showed you today, they've done a wonderful attempt and successful attempt at trying to translate your rat work into human work. So I'll give you a lot of kudos for that. Other questions in the room or on? Yeah.
Well, I know this research was done primarily involving like alcohol abuse. Can this be translated into drug abuse and the effects of like exercise relating to drug abuse?
Did you hear that, Leigh? The question is, can you translate alcohol, switch alcohol with the word drug abuse?
Yes. So exercise seems to repair the brain damaged by any substance. What's really interesting, though, is that there's an important difference between alcohol use disorder and exercise and substance use disorder and exercise. And that is that there is a positive relationship between physical activity and alcohol consumption. So such that the more active people are, the more they tend to drink. And so I've always thought this is an important thing to take into account if we're going to tell people recovering from alcohol use disorder that they should go exercise. And so one of the things that my colleagues and I have been trying to do is figure out, well, who exactly are these people that are both exercising a lot and drinking a lot? And whatever is going on with them, does that apply to people who are recovering from alcohol use disorder? Because yes, exercise might repair the brain after it's been damaged by alcohol, but do you want to run the risk of people drinking more when that's exactly what they're trying not to do? So, so to answer your question, exercise appears to be very good for repairing the brain after it's been damaged by any substance use disorder. The difference is that no other drug has a positive relationship with exercise such that you take in more of the drug if you exercise. It's exactly the opposite for everything but alcohol.
That really is interesting. Makes you really think about what is it about alcohol itself that those who exercise and exercise a lot tend to then want or drink more alcohol. Sorry, my brain's working overtime right now. That's really interesting, trying to figure out how you would even test that, but that's a different conversation. Other thoughts, questions? Yeah, Carolise.
I was thinking, is there a difference in like beneficial effect between moderate to high intensity workouts, or is it just exercise in general?
Did you hear that, Leigh? Is there a difference? Does the intensity of exercise matter?
It depends on what you're looking at. For example, if you're looking at the effects of physical activity on hangover, then yes, it has to be vigorous physical activity. But if you're looking more broadly at how much exercise do you have to do in order to benefit the brain, like let's just say the perfectly healthy brain, no neurodegeneration, no pediatric radiotherapy, no binge alcohol, how much do you have to do to benefit your brain? You don't have to run a marathon. I think there's good there's good data that walking will do it. And as I said earlier, it's whatever you will do. Whatever you will do on a daily basis, whatever your preferred type of exercise is, do it and it will benefit your brain.
Yeah, there's a big push, right? Get up for 5 minutes and go do something. Even if it adds up to, I guess the new number, these numbers change all the time, is 20 minutes, right? If you can get 20 minutes of activity a day, that's supposed to help X, Y, Z, and K. So that's why if you take my class, I try to get you all to get up and move for 5 minutes and like get out of the room or get off of Zoom. So I helped your brain just a little bit.
I love that. Tell me about this yoga class that was part of Brain Awareness Week.
Yeah, so NeuroSci is a national neuroscience society, honor society, and every year they have a theme. And this year's theme was the effects of exercise on the brain, which is why you are here, my friend. But Maya Holman, who is, I'm not sure if she's still online, she's not feeling well today, but she is there on Zoom. She's our president. She wanted us to actually say, okay, well, we're not just going to talk about exercise in the brain. Let's go actually and do something. So she organized a yoga/bar hybrid class at the Ray, which is our exercise facility. So that was day one of Brain Awareness Week.
I love it. I am a certified yoga instructor, and I'm an N of 1, but I believe very strongly in its benefits for my brain.
Any other questions in the, yeah?
Do you speculate that the benefits of the exercise, or like especially in relation to alcohol, could potentially be due to the intake of like oxygen? Like, does that make sense?
It's something about oxygen.
Is there a relationship between the effects of exercise on the beneficial effects of exercise? Does it have something to do with oxygen? Is that what you're saying, Ravinder?
Yeah, like oxygen intake.
Is it because you're bringing in more oxygen while you're exercising? Is that what you're saying?
Yeah.
Well, bringing in more oxygen is good for the brain. It will, keep it healthy, make sure that it's getting, the blood supply and all the good stuff that it needs, so that when it does get hit with something like a binge, it has the reserves and the infrastructure to withstand it. So certainly depriving a brain of oxygen is known to make it more vulnerable to future insults. So I think Dr. Kozlowski could actually talk about that from her work with hypoxia and domestic violence. So let's just put it this way. Making sure the brain has enough oxygen and oxygen being in good supply is definitely going to help protect it when it does encounter an adverse situation.
Yeah, absolutely. Our brain requires a lot of energy to work, and part of that is receiving glucose and such, but also part of it is receiving enough oxygenated blood, etc. Okay, I want to be mindful of time. Anything else on the chat?
I don't think so.
Okay, no. All right, one last question in the room. Yeah, Carolise.
Yeah, can the endorphins rush be contributing to the effect?
Do endorphins play a role in making the brain better after exercise?
It certainly plays a role in you feeling good after exercise. So, no, seriously, I ask this of classes all the time. I say, okay, who likes to exercise? And some people will admit to it, and I say, okay, who likes how they feel after exercise? And then everyone's like, oh yeah, right. So there's the idea that maybe while you're doing the exercise, you're maybe not loving it, but you feel pretty good afterwards. And so the endorphins are definitely contributing to that.
Yeah, that's me. You have to drag me to do it, but once I'm doing it, I feel okay. Okay, well, before you go, just wanted to let you know, this is in the mail. We're gonna put this in the mail for you. I don't know if you can see me with it. You'll be an honorary member of DePaul's neuroscience program. So stay tuned for that. But please join me in thanking Dr. Leasure again for coming and finishing off Brain Awareness Week with us. Thank you. Thanks so much, Leigh. I will be in touch soon. Thank you.
Okay, bye.